Hello everyone. So we have already discussed about gram-positive cocci, and we have seen how these gram-positive cocci can be classified and identified under a microscope based on what their morphology or shape is on gram staining and in microscope. So in continuation with that, today we are going to discuss a very important group of gram-positive cocci, that is Staphylococcus species. So today we'll be going in depth and in detail about this Staphylococcus species. So in this session, we will basically understand the morphology as well as learn about the culture characteristics of Staphylococcus species. We will also get to see a few of the biochemical reactions that happens in this species so that their laboratory identification can be done. And then we will look into various species of Staphylococcus that are out there and what is the clinical importance of each of these species. So staphylococci are gram-positive cocci. Gram-positive, why? Because they are purple in color when we stain them using a gram stain. And cocci because they are spherical in shape. So because of their shape, they are round in shape, we call them as cocci. And because they are purple in color, we call them as gram-positive. So it is clear by now that staphylococci are gram-positive cocci. Now we will look into the arrangement, how these cocci are arranged. Actually, what happens when these organisms divide, they divide in three different planes, like in multi-plane manner or in a multi-directional manner. So they will form bunch of clusters like a grape. As you have seen in grapes, there are bunch of cocci or clusters that is seen. Similar is the arrangement in case of a Staphylococcus species. So this name Staphylococcus was actually given by a scientist, Augustin, from a Greek word staphyle. Staphyle means bunch of grapes and cocos means berry. So because of their morphology and how they appear under microscope, they are called as Staphylococcus. Means gram-positive cocci arranged in a bunch of clusters, in a bunch of grapes. Now, second thing, they are ubiquitous. What does this mean? Ubiquitous means it is present everywhere. It is present in the environment. It may be present on your skin. It is the normal flora of our skin and mucous membrane. Skin as well as mucous membrane. Mucous membrane means like throat or any other area which is nearby the skin. So these are ubiquitous, they are present everywhere and they can be a part of normal flora, means they are the commensals on skin. They can be normally present without harming you. So they are present on skin as well as mucous membrane. But why we are studying them and what is their importance in a clinical setting is that some of them can cause opportunistic infections. Opportunistic means they are normally lying there on the body, but once they get the opportunity, they can cause you infection. Now, what is this opportunity? This opportunity could be like you are taking any antibiotic which wipes out the sensitive organism. So the resistant one, more hardy one will survive. And most importantly, if somebody is on immunosuppressants or anybody's immunity is depressed, at that time these organisms can take the opportunity and cause frank infections, especially in hospital environments. They are lying there, they are surviving there in the environment, but once a patient comes or if the immunity of the patient is lowered, they can get the opportunity to cause infection. So you can call them as opportunistic infections. Suppurative infection. Suppurative means the infection that create pus formation. Any infection where pus is formed, we call them as suppurative infections. Staphylococcus aureus is especially notorious and known to cause such kind of suppurative infections or pus forming infections like abscesses. So morphology is quite clear by now that these are spherical cocci, gram-positive cocci. What is their size? They are about 1 micron in diameter. Means when you look at the cocci, this diameter is about 1 micron. Okay. Arrangement, we already know it is grape-like clusters. They are gram-positive, means purple colored. They are non-motile. So some bacteria move, some bacteria do not move. So those who do not move, they are called non-motile. Same is the case with Staphylococcus. It falls under the category of non-motile organism. It will not move. Non-sporing, we know few bacteria, they make kind of spores as a mechanism to survive under difficult conditions. So if this is the bacilli, this structure, that is round structure, is the spore. like bacillus or clostridium. You will see in these organisms they form spores, but staphylococcus is not a spore forming organism, so we call them as non-motile and non-spore. Now we have already seen how they divide. They divide in multiple planes. Means it is not one dimensional, it is not two dimensional. It is in multiple planes. They will divide and form cluster-like arrangement. So microscopically, they are purple colored cocci. Pus samples may show abundant neutrophils with intracellular cocci. If you see a pus sample under the microscope, these staphylococci may be present where there are abundant neutrophils. Neutrophils are blood cells, white blood cells that fight against infection. So these neutrophils accumulate in the pus to fight the infection, and intracellular means within the cell, within the neutrophil. Suppose this is a neutrophil, you can see cocci within the cell. We will see it later on. Occasionally capsules seen in young culture, but this is not very common with Staphylococcus. So here you can see a microscopic slide. This slide is made from a bacterial colony, which means you have cultured some sample in which Staphylococcus infection was there on a plate, like blood agar plate. So we have cultured the organism on this plate, and from this tiny colonies develop on culture media, and this colony is picked then to make a smear or slide. Okay, so this slide will show you this kind of a picture. So this is a colony smear in which you can see various purple colored cocci, and they are arranged in bunches or clusters. There are multiple clusters you can see. So this is how they appear. The colony appear under a microscope. The smear from a colony. Now this is the actual sample which we were discussing. This is a smear from a pus sample, and this bigger pink thing that you are seeing, this is a pus cell or a neutrophil. Some of them are lymphocytes, but mostly they are the neutrophils. Like here, you can identify it as a neutrophil. It is a bilobed, and lobes you can identify here. There are also multiple lobes in these cells. So these are basically neutrophils. So there are abundant neutrophils, many neutrophils in the smear. And then you can identify there are some purple colored structure. This is a cluster of gram-positive cocci. So this is an actual sample from a person who is infected with the Staphylococcus species, and here you can see the organism is seen intracellular. Now you understand what I was telling. Intracellular means within the cell. So this is the neutrophil, and within the neutrophil you can see Staphylococcus. So these are intracellular organisms. Sometimes they are seen inside the cell, sometimes outside. This is again a bunch of Staphylococcus. So now let's look at the culture characteristics of this organism. So basically we grow the organism on some solid media, like in this case, this is a blood agar plate. On blood agar, we have processed the sample of a patient who was suffering from Staphylococcus aureus. So these are the colonies that we got from Staphylococcus infection. Now, what kind of environment these organisms need to grow? So these organisms are aerobic and facultative anaerobic. What does this mean? It means they are aerobic. Aerobic means they grow in the presence of oxygen like any of us. So we all need oxygen to grow. Similarly, this organism will also need oxygen to grow. So anyone who needs oxygen to grow, any organism, we call them aerobic, means they want oxygen. But the special feature of this organism is that they are facultative anaerobic also. Now, what does this mean? It means anaerobic means growing in the absence of oxygen. So this bacteria by nature does not grow under in the absence of oxygen. It needs oxygen. But if need arises, if it does not get enough of oxygen, then it can also survive under those conditions. So they are called facultative anaerobic. Like by nature, by default, they are aerobic. But if they don't find oxygen in some place, they can also grow on these places. So these are facultative anaerobe. Like in an infection where pus is formed, we call them abscess. So there are pockets of pus. In pus, the oxygen content is very low. So in these conditions, this bacteria will use this quality and grow and survive and cause infection. So they are aerobe, and facultative anaerobe means they require oxygen, but they can also grow in the absence of oxygen. Now, once you process these samples, like plate this sample, you want to grow the organism, what kind of temperature you need? You need 35 to 37 degrees Celsius. These plates are kept in an equipment which we call as incubators. So in incubators, you keep these plates and place them 16 to 18 hours at a temperature that is 35 to 37. Optimum means the best temperature or the temperature at which the organism grow most or best. So this is the best temperature to grow this bacteria, which is 35 to 37. Now we will look how these colonies look. So this is a confluent growth, heavy growth. If you see in lesser growths, there are circular, convex, smooth colonies in this bacteria. Convex means they are dome shaped, this kind of colonies. Circular means round. Smooth, you can see in the picture, they are smooth colonies. And opaque means they are not translucent, you cannot see through the plate under these colonies. So they are smooth, they are convex, they are circular, and they are opaque. Many strains produce golden yellow pigment. As you can see in the picture, they are not typically white colored colonies, they are slightly yellowish colonies. This is a specific feature for Staphylococcus aureus. This will not be seen in other species of Staphylococcus. So, in Staphylococcus, there are multiple species like Staphylococcus aureus and Staphylococcus epidermidis. In Staphylococcus aureus, you will see this golden colored pigment. Okay, but in Staphylococcus epidermidis, you will not find this golden colored pigment and the colonies are white colored. Growth on medium. So you can also use, apart from blood agar is good for growing. Nutrient agar is can also be used for growing these organisms. Again, they form large, slightly pigmented or yellowish colonies on a nutrient agar slope. Slope means in a tube or in a small vial, you make the media like this. So on this media or slants, when you plate the organism, the colony appearance is known as oil paint appearance. Means it is oily in nature. Pigment. Now we see that golden yellow pigment is produced by this bacteria is non-diffusible. What does this mean? This means that if the organism is grown on a plate or on a tube, this pigment will not go into the media from the colonies. It will stay in the colonies. This will not diffuse. When you will be looking in Pseudomonas case, the pigment is diffusible. It goes into the media, it diffuses into the media. If it is a liquid media, it will turn completely greenish or bluish. So the Staphylococcus, this is opposite. It is a non-diffusible medium, which means it will not leave the colony and go into the solid or liquid medium. And this pigment is produced best at 22 degrees centigrade aerobic culture. So if this condition is given, the pigment is enhanced or best formed. On blood agar plates, you can also see some beta hemolysis. What is beta hemolysis? When it is grown on a blood agar plate, suppose there is a colony, around these colonies there is a zone of hemolysis, that is clearing of the media. The media will appear lighter in color and it will clear out. So commonly seen in sheep or horse blood agar. These two agar plates, the hemolysis is best seen. Another medium is mannitol salt agar. Mannitol salt agar is a selective medium. What does selective medium means? Selective medium, as the word indicates, means that it will select or prefer the growth of Staphylococcus and it will inhibit other organisms. So if in a sample there are a lot of organisms, many organisms, and you especially want to see if Staphylococcus is there, then mannitol salt agar is a good medium because it will preferentially select. Why? Because it has some salt. Salt is preferred by Staphylococcus aureus, so they will preferentially grow. Mannitol fermentation gives yellow color colonies. Since this media has mannitol, Staphylococcus does what? It will use that mannitol. It will ferment that mannitol. And once this mannitol is used, yellow colored colonies are formed. If they are not fermenting the mannitol, pink colored colonies will be formed. Okay. High salt tolerance. Since Staphylococcus has high salt tolerance, it will grow under high salt concentration, 7.5 to 10% NaCl. It can tolerate, it can grow. So these are few selective media you can remember. Mannitol salt agar, salt milk agar, Lutlam's tellurite media. And high salt concentration inhibits most other bacteria but not Staphylococcus, so you will select Staphylococcus. Biochemical reactions are very important. We will learn about them in detail in a separate class. But here you can understand the basics of biochemical reactions, like what biochemical reactions are shown by Staphylococcus. First test, the most important test, is a catalase test. Catalase test is done using a chemical called H2O2. So we take on a slide H2O2, a drop of H2O2, and then we touch the colonies of Staphylococcus on this H2O2. In case of Staphylococcus aureus or Staphylococcus species, bubbles are formed, which we call as effervescence. So, if bubbles or effervescence is forming in catalase reaction, it is catalase positive. Staphylococcus species are catalase positive. Coming to the coagulase test is also a simple test. It can be a slide or a tube coagulase test. Like in a slide, you emulsify colonies of Staphylococcus aureus or epidermidis, whatever you are going to test, and you take a drop of a serum. Okay, and then mix these two drops. In case of Staphylococcus aureus, clumps will form inside the serums because it will coagulate it. So, coagulase test positive is a special feature for this species, that is aureus. Coagulase test will be negative in Staphylococcus epidermidis. We will see the difference later on. So, this is a very peculiar feature for Staph aureus. Catalase is a feature for all Staphylococcus species. Coagulase is a feature of Staphylococcus aureus. Urease, again, this is a test biochemical reaction shown by Staphylococcus. Nitrate reduction, again a biochemical test, positive in this. Gelatin liquefaction also seen. MRVP test are positive and indole test is negative. The details of this test we will discuss separately when we will be discussing about the biochemical reactions. So Staph aureus versus epidermidis. Broadly, if you want to see what is the difference between the two important species in these groups, Coagulase we have seen it differentiates Staph aureus from epidermidis. Mannitol fermentation we have seen the colonies become yellow in color when they ferment mannitol. So it is again a case of Staph aureus. Beta hemolysis, the colonies are usually hemolytic, means they clear out the area around themselves on a blood agar. Heat stable nucleus is produced by Staph aureus. Golden yellow pigment we have seen is produced by Staph aureus. Lysostaphin sensitivity means this is inhibited by lysostaphin. So all the features will be seen in Staph aureus, but all of them are negative in Staph epidermidis. Out of this, coagulase is the most important characteristic test to differentiate the organism into Staphylococcus and other species which we collectively call as cons. Cons means Coagulase negative Staphylococcus species, and Staph epidermidis is one of them. So they are basically two type groups you can consider. One is Coagulase positive, one is Coagulase negative. Staph aureus is Coagulase positive, whereas Staph epidermidis is Coagulase negative. So we will be discussing about mainly three important species. First is Staph aureus. We have already seen. It is the major human pathogen. We have seen it causes pus forming infections, abscesses. The another one is staph epidermidis. Epidermidis is most commonly, as the name suggests, present on the epidermis, on the skin as a commensal. But when the opportunity arises, if the patient is admitted, if there is any device and it gains the entry, then it can cause device-related infection, like you have put a catheter line or any IV drip, anything, any breach in the skin happens. So they are, since they are present in the skin, if you don't follow the proper technique of inserting a catheter, then they can gain entry into the bloodstream and cause device-related infections. Third is the staph saprophyticus, again a part of coagulase negative staphylococcus. One is the staph, these two are cons. So Staphylococcus saprophyticus is an important cause of urinary tract infection among females. So let's come to the key take-home points. We know now that these organisms, that is the Staphylococcus, they are arranged in grape-like clusters. Staphylococcus aureus produce golden pigment on media and also causes beta hemolysis when we grow it on a blood agar plate. Coagulase test is most important key differentiating test, positive in Staph aureus and negative in the other most of the Staphylococcus species, which we call them as cons. High salt tolerance is again a characteristic feature, and this feature is used to make selective media to grow the organism selectively. Now we are coming to the MCQs. Staphylococcus can be described as gram-positive cocci in chains, gram-positive cocci in pairs, gram-positive cocci in clusters, or gram-positive bacilli in clusters. Which of these is correct? You can describe Staphylococcus as gram-positive cocci in clusters. So this is very simple now. Coming to the question number 2. Cocci in Staphylococcus divide in one plane, two planes, three planes, or multiple planes. Yes, it is multiple planes. They divide in multiple planes. That's why they form a bunch like cluster. Okay? For a cluster to form, for a bunch like a grape to form, they have to divide in multiple planes. MCQ number 3. Staphylococci are strict anaerobe means they will only grow in the presence of oxygen. Strict anaerobe means they will only grow in the absence of oxygen. Strict microphilic means they only need a very minimal amount of oxygen. Micro means small amount of oxygen. Or they are aerobe and facultative anaerobe means they can grow in oxygen but can also survive without oxygen. Yes, it is aerobe and facultative anaerobe. Question number 4. Tests that can differentiate Staph aureus from Staph epidermidis are all except. So mind it, here is except. So 3 of the tests will differentiate between the two, but one of them will not differentiate. Which test is that which will not differentiate? Coagulase test, Catalase test, mannitol fermentation, and beta hemolysis. Catalase test. Catalase test is positive in all Staphylococcus species. It is used to differentiate between Staphylococcus and Streptococcus because Streptococcus is negative in catalase while Staphylococcus is positive. But when you have to differentiate between the species, then coagulase, mannitol, beta hemolysis is used to differentiate between aureus and epidermidis. Okay. Now select the best explanation for the image below. Below you can see some image and we will see what is the explanation. Just see the image nicely. There is something lying here and here. This is colony smear of Staphylococcus, means this smear is made from colony which is grown on blood agar. Colony smear of Streptococcus, this is a smear which is made from a colony grown on a culture medium. Gram stain of sample with few pus cells and gram negative bacilli. Is it a sample which has few pus cells and GNPs? Or is it a gram stain of a sample with abundant pus cells and gram positive cocci clusters? This is again a very simple thing. Yes, it is a gram stain of a sample showing abundant pus cells. This is a pus sample with lots of pus cells. Pus cells are nothing but WBCs and gram positive cocci. Thank you very much. Please subscribe to the channel if you like the video. See you again.